everyone, good evening. Welcome to Aakash, BYJU'S NEET channel. I'm your teacher Ankita and I welcome you all in today's class. I hope that all of you are doing really great and are excited for our today's class. In today's class, we will be discussing about the DNA replication. It's a very important topic. Various years we have seen that we have the question coming from this particular topic, so I hope that all of you are ready. Good evening everyone. Do let me know if you can see me and can hear me clearly. Good evening Manas, Naruto, Shalini, Lavanya. Good evening everyone. How are you all? And how's the Josh everyone? I can see so many of you here. Good evening everyone. Please make sure you stay with us till the end. It's a very interesting topic and we will try our best to finish it off really very quickly, right? Yes. Good evening everyone. Good evening. Hello. Selvanti, Manas, Naruto, Roshni. Good evening. I hope that all of you are good. Yes, I'm doing good. Thank you for asking. Hello Rajkumar. Good evening. Thank you Lavanya. I'm doing good. Okay. So everyone, in today's class, we know that what we'll be discussing about the DNA replication. Right, of course, apart from that, we'll be looking at the semi-conservative principle also, that how we saw the proof that DNA is semi-conservative in nature. So we have a lot of the important topics today. So without taking much of your time, everyone, let's get started. This is the part 4 that we are in, that we'll be doing in today's class, right? It's a part 4. And we have done 3 parts already over here. I have the images. So everyone, if you're new here, it could be that you're watching this session later. It could be that you are a new subscriber on the channel. Please make sure you go and watch these videos. We discussed about the structure of the DNA. Then we discussed about the packaging of the DNA, the search for the genetic material. In today's class, we're discussing about the DNA replication. So all of you, are you there with me? A quick thumbs up, all of you. A quick thumbs up and a quick smiley in the chat. Good evening, everyone. Good evening, Pawan. Thank you so much. Ashwini, Nabin, Bismita. Okay, done, right? Awesome. So everyone, in the last class, I asked you these 2 questions, right? So we have the question which is, this is question number 1 and that was question number 2. By performing a series of experiments that show the effect of RNS strains of Streptococcus pneumonia on mice, Griffith concluded that, yes, what he concluded? He concluded that very good, yes, option number D is a correct answer. The non-virulent bacteria were transformed by the heat-killed bacteria. Absolutely correct. So here we have the correct answer for it. And then of course, viruses grown on a radioactive sulfur contain what? They contain radioactive protein but not DNA. So that was the 2 questions, everyone, I gave you as a homework in the last class. Good. Right. I will be calling out your names in the later part, right? There are so many students who gave us the correct answer. So very good, everyone. Okay. So everyone, are you ready for the class today? You can just change the setting of your phone if the screen is not that clear. Okay. So let's get started, everyone. Now in today's class, we'll be starting, of course, we'll be looking into the DNA replication, and of course we'll be looking into that how it got proved that DNA replicate in a semi-conservative way. So let's understand that. Now if I ask you, how does this DNA divide? So we know that DNA is present inside the cell, right? We saw its packaging. We talked about that, how the search of genetic material actually happened by the series of experiment done by various scientists, right? And after a long research, right? After a long research, yes, what we got as a conclusion that, yes, DNA is a genetic material, right? So of course we go back and we talk about the first experiment done by Griffith in the year 1928. Then of course in between we have the biochemical analysis for it that took approximately 10 years from 1933 to 1944, right? We have 3 important scientists. We have Avery, McLeod, McCarthy, right? And then of course we discussed about the Hartley and Chase experiment, right? So that gave the conclusion which happened in the year 1952 that DNA is a genetic material. And then we have Watson and Crick model in the year 1953. So these are the things that we have. And Watson and Crick actually said that, DNA replicates in that way. Yes, we have discussed this, right? They clearly said that DNA replicates, right, in a semi-conservative way. but there was no proof of it, right? They didn't went out and said, yeah, we have the proof of it. So we will be looking into the experiments, right, that actually proves. So we'll be talking about it. So let's get started, everyone. Before we start, we should know what is replication. It's a process of copying and duplicating of the genetic material. The DNA is the, it's a very simple term. We have the replication, and of course, we have a very important experiment done by the Meselson-Stahl right over here in the year 1958. Super important experiment, everyone. Yes, Bhavan 451 was discovered by Franklin. Right? Rosalind Franklin. Okay. Along with the Winkling. Okay. Everyone, are we clear? Both of them did the X-ray diffraction method, right? They have that performed and we have that image. Okay. So now everyone, let's get started, right? Very good. Very good. Yes. Okay. Chaliye. So we have Matthew, Meselson, and then we have Franklin-Stahl, right? And of course, they did their experiment on the E. coli. So what they did? Everyone, what they did? Let me just quickly give you a quick summary of what they did, and of course, then we'll be looking into it over here. So these two scientists, right, did an experiment on the E. coli, right? Yes, what they did in this particular experiment, they actually grew E. coli on NH4Cl, right? And basically they added the heavy isotope of nitrogen, right? So everyone, are we clear with this? Clear? So we have this important thing that we have over here. Yes. Hello, thank you so much. Nice, very good, very good. Yes, everyone, so are we clear with this? So it was an isotope, right? It was an isotope of that. Clear? Now, we have what we have 15. So ये सबको clear है, right? And of course, after the 20 minutes, what we saw, of course, the replication will happen, right? And of course, they will divide. And what they did after that, they did the centrifugation. Right, and in the centrifugation, we know that as the nitrogen got incorporated inside the DNA, we saw that the DNA was there at the bottom because it's a heavy nitrogen, right? It's not 14, but it's 15. So we saw that the nitrogen was at the bottom. And in the centrifugation, what we use CSCl, especially where we can separate it out. We have this a fluidy thing, and of course on the other side we have the DNA. So for the separation, to maintain the different concentration gradient, we use this, and that's actually help us to see it. Right? Now we did this a similar experiment. What we did, we take these, right? We took this E. coli and we actually took them to a new petri dish which has N14. Now what we have N14. And after 20 minutes, like if you have 20 plus 20, right, that will give us a 40 minutes. So after this, what we saw something very different, right? We saw that somewhere we have our DNA in between. It was not at the top, it was not at the bottom, it was there in the between. Everyone, are we clear? Yes? Everyone, are we clear? We saw this, right? And based upon these experiments, we did a one more set, and that's how we got the conclusion. So everyone, do you have a little bit idea what this experiment will be about? Yes? Clear? So this is just a brief, it's not a complete experiment. We will be looking into the details, but अब देखते हैं, चलिए. So now we know that what was the step one? Step one क्या था? We grow the E. coli in the NH4 and media में, जहाँ पे we have heavy isotope. Nitrogen क्या है? 15 है. It's a heavy isotope. Then of course we grew it, the E. coli on, right, in that particular medium. And of course the E. coli will take the nitrogen that is growing on. है ना? Nitrogen है यहाँ पे, तो E. coli उस nitrogen को ले लेगा, which is very obvious. उसके बाद में क्या हुआ? Let's quickly recall what are isotopes. What are isotopes? Here. Those of you who have any confusion, you can take a screenshot over here. Isotopes में क्या होता है? Atoms of the chemical elements having a similar atomic number, but they have a different atomic mass. Basically, they have a different mass number. So here we have, okay, a quick recall, a very quick recall of the isotope. Then we know that N15 is a heavier isotope of nitrogen and not the radioactive. Radioactive isotope we have discussed in the last experiment, right? The experiment which was done by the... Can you quickly tell me which experiment we're talking about? Right? हमने कहाँ पे ये experiment देखा था जिसमें radioactive isotopes use करे थे? Phosphorus 32 and sulfur 35 किस experiment में use किया था? जहाँ पे we use the bacteriophage to infect the E. coli, and this particular experiment proved that the DNA is a genetic material. Done by two very famous scientists, very good, Herschel and Chase, absolutely correct. तो ये heavier isotope है, it's not the radioactive isotope. Clear? Then of course what we did, here we have the picturization, right? We have a very clear image of the DNA with the 14 isotope, and of course over here we have with the heavy isotope. So that's a major difference, everyone. Okay, once we are done with that, we move to the step number 2. And what is the step number 2? We will transfer these E. coli with the heavier nitrogen into the normal. Now when we have this E. coli which has nitrogen 15 into the medium which have nitrogen which is normal. Everyone, are we clear? क्लियर एवरीवन? हमने कहां से स्टार्ट किया? हमने स्टार्ट किया 15 नाइट्रोजन से, जो हैवी आइसोटोप है. पहले उसे ग्रो किया, वहां पे रेप्लिकेशन हुई, वहां पे रिप्रोडक्शन हुआ, इकोलाई ने रिप्रोडक्शन किया वहां पे. उसके बाद में अब कुछ सेल्स को हमने डाला नाइट्रोजन 14 में. एवरीवन, आर वी क्लियर विद दिस? दैट इज द सेकंड स्टेप. मीडियम, वी हैव चेंज द मीडियम over here. Now what will happen? After some time, after 20 minutes, after 0 minutes, after 20 minutes, and after 40 minutes, we took the sample. Now what are we doing? We are taking the sample and we'll be doing the process of centrifugation. Now we are using the CSCL centrifugation for the DNA sample. And again a very important question, that why we use this? We know that CSCL, what is the meaning of CS and Cl over here? Yes, I think many of you said it earlier also. What this CSCL stands for? Yes, jaldi se everyone. Very good, calcium chloride, right? Very, very good. So, this particular technique of centrifugation actually helps us in the separation of the particles on the basis of size, shape, density, and the viscosity of the medium. That is the reason we are using this CSCL over here for the process of centrifugation. So once we did that, we isolated the DNA samples, right? And we added this, and of course we saw the process of centrifugation happening. Centrifugation कर रहे हैं. यहाँ तक सबको समझ में आया? मैं दोबारा जल्दी से बोलती हूँ. अब हमने क्या किया? हमने एक पेट्री डिश पे N15 जो हेवियर आइसोटोप है नाइट्रोजन का, उसके साथ में इकोलाई को ग्रो करवाया. तो वो जो नाइट्रोजन था इकोलाई ने एब्जॉर्ब किया, उनके डीएनए में आ गया. अब हमने क्या किया? वही जो इकोलाई के पास में हेवी नाइट्रोजन का जो आइसोटोप था, वो जो थे ना उनके पास में, उनको हमने नॉर्मल नाइट्रोजन मीडियम में ग्रो किया, जो अब N14 है. ग्रो किया, उसके बाद में हमने डीएनए सैंपल्स कलेक्ट करे और क्या प्रोसेस किया ऑफ द सेंट्रीफ्यूगेशन यूजिंग CSCL. एवरीवन क्लियर? यहां तक सबको समझ में आ गया? क्लियर एवरीवन? ऑल ऑफ यू गिव मी अ क्विक थम्स अप. Yes everyone, are we clear with this? Very, very good. Everyone, please make sure you stay with us till the end. थोड़ा सा difficult topic है, but don't get afraid, it's easy peasy. चलिए. Now, after the centrifugation, what we saw that there are various bands, right? Can you see these bands? These bands appeared on the centrifugation. Let's understand about these bands. So first band talks about, right, the band which is there at the lower side of the bottom, basically at the bottom of the test tube, right? At the sample at the 0 time, what we saw, heavy band is there. Of course, when we added the nitrogen, right, which is heavy isotope, N15, it's a heavy isotope. जब हमने वो add करा तो क्या होगा? Of course, जो DNA sample है हमारा heavy है और वो एकदम bottom पे है. After 20 minutes, जब हमने उनको N14 के medium में ट्रांसफर कर दिया था, so what will happen? Again, जब यहाँ पे ट्रांसफर किया तो we saw few different things happening. Now let me explain you over here. So we know that 15 था, Yes, it is 15, 15. अब हमने क्या किया? अब हमने V ट्रांसफर इट टू दी N14. नाउ येलो भी है, ठीक है, और हमने N14 भी दिया है. क्लियर? एवरीवन आर वी क्लियर? तो ग्रीन वाला क्या हो गया? N14 हो गया और येलो वाला इज N15. तो यहां पे क्या आया? Hybridization. We saw the hybrids. Yes? Okay, everyone? Clear? So what we saw over here? We saw it's a hybrid. Done, right? यहां पे क्या हुआ? 15 आ गया. When we change the sample, N14 में जब करा तो we saw N14 का भी DNA है, right? वहां से भी उसने नाइट्रोजन एब्सॉर्ब किया. So DNA में हमारा N14 भी है. So यहां से हमें एक चीज पता चली कि दोनों ही strands आ रहे हैं. Clear, everyone? It's a mix of both the strand. Both the strand in the sense, both the nitrogen 15 and nitrogen 14. दोनों ही हैं. तो यहां पे हमने क्या देखा? We got the hybrid. जो बैंड कहां पे आया? बीच में आया बैंड. Clear? And after the 40 minutes, what we saw? We saw two bands of the light, and of course we have the hybrid band. Everyone, are we clear up to here? Thank you, sir. Everyone, are we clear up to here? Jaldi se. Yes? Clear, everyone, are we clear? Okay, chaliye. Now that we are clear with this, let's understand this important thing. N14 hai, right? We have N15 also. Clear? So I'll make the N14 and green color. Okay? And we have this. ठीक है? अभी तक ये समझ में आया ना हमारे पास में? After 40 minutes what changed? क्योंकि NCERT में दिया है 20 plus 20 40 हो जाएगा, इसलिए we're using this 40 minutes over here. ठीक है? After 40 minutes what will happen? Now, the important thing for all of us to remember is कि ये जो है यहाँ पे N14, of course, it will act as a template and of course इसका एक duplicate होगा. We'll have one more strand इस तरीके से. अब ये जो strand है किसका होगा? It will be similar to the N14. यहाँ पे हमारे पास में N15 है, right? वहां पे what we'll see? We'll see that it is of N14. So we have the mixed breed also, and of course we'll have the N14. Everyone, are we clear? Yes, कोई confusion तो नहीं. Please do ask your doubts if you have any confusion, right? Yes, chaliye. Now, if you're clear with this, let's quickly see this how it looks. So at the heavier band, at 0, we have this. We have both the DNA, right? Sorry, we have the DNA which have isotope 15, which is a heavy DNA, right? We can see both the strands have nitrogen 15, which is the heavier isotope. After 20 minutes, we saw that we have a hybrid. What we have? We have the hybrid. We can see we have N14 also and we have N15 also. Then at the last, everyone, at the 40 minutes, what we saw? We saw something very, very interesting. Both the strands have N14. See over here, we have our strands, we have a DNA that has, both the strands are N14. Again, it clearly shows that it is just conserving the semi, right? Clear, everyone? Yes? We saw the semi-conservative nature of the DNA. पूरा कॉपी नहीं हो रहा है, आधा ही हो रहा है. तो फर्स्ट 20 मिनट्स के बाद में जो हमारे पास में जो N14 था उसने अपनी दोबारा से कॉपी बनाई and we have a strand which is light band. Clear, everyone? And we have the hybrid also. Clear? इतना clear है? So अगर हम स्टार्टिंग से देखेंगे from 0 to 20 to 40, we can see that it is a कुछ कंजर्व हुआ है. 15 अगर था स्टार्टिंग में तो अभी भी लास्ट में है हमारे पास में. Yes or no? कंजर्व हुआ है, पूरा नहीं है पर कंजर्व हुआ है. Clear, everyone? This is nothing but was the proof that the DNA replicates semi-conservatively. मतलब पूरा change नहीं कर रहा है, एक ही strand है हमारे पास में जो नया add हो रहा है. Clear? So we have, initially we have the heavy band, we have the hybrid band, and of course at the last we have both. We have the light band and we have the hybrid band also. Clear everyone? Thumbs up if you're clear with this, right? So you can take a screenshot of this everyone. Jaldi se you can take a screenshot of this. Very good everyone. Do take a screenshot of this. है ना? तो first generation, second generation और हम इस तरीके से याद रखेंगे. So it actually help us to understand the semi-conservative DNA replication. DNA क्या करता है जब अपने आप को replicate कर रहा है? तो जो parental DNA है वो दो parts में हो गया. Half पुराना होता है और half new बनाता है वो strand. DNA अगर ऐसा है, right? DNA अगर ऐसा है, अगर ये open होगा तो ये strand हो गया और ये हो गया. And हम क्या देखेंगे कि DNA एक नया strand बनाएगा. So it is semi-conservative. Half है ना, full नहीं है. See, I'm sure you will agree. This is half only. It's not full. पूरा exactly copy नहीं किया है. दोनों strands ऐसे नहीं के साथ में आ गए. It's a different strand, right? एक नया strand बनता है based upon the parent strand. Everyone, are we clear? भास्कर, we will discuss about it कि new DNA कैसे बनता है. Clear? We'll discuss that in the DNA replication. Everyone, are we clear? Yes, chaliye. So that's a part. Now we'll be looking into the DNA replication. So everyone, are we clear? Quick thumbs up everyone. जल्दी से बता दीजिए. Good, very good. जिनको doubt है, please don't worry, हम एक बार revise करेंगे, so you'll be able to get it. And if not, you can watch the session again. Okay. Now we know that these two scientists, Meselson and Stahl, proved that the DNA replicate with the or by the semi-conservative. The DNA replicates in a semi-conservative way in the prokaryotes. So they worked on the prokaryotic E. coli. Whereas, if you talk about in the eukaryotes, do you think we have something? Absolutely, right? So the Taylor, right along with his team, worked on the eukaryotic cell. And which eukaryotic cell he worked upon? He worked upon the cell which is of the broad beans, right? So here he worked upon the bean plant. It's a plant. It's a eukaryotic cells. Okay. So basically we saw the experiment happening, right, on both the prokaryotic cells as well as there on the eukaryotic cells. And both of these concluded that the DNA replicates in a semi-conservative way. Clear? Clear everyone, done? Chaliye. If you're done with this, let's talk about the process of replication. Shalini, that was a homework question I thought. If you want the answer, we'll just give you till the end of this class, but you can find out more about it. सोचो क्या होगा? Conserve होगा या नहीं होगा? The time will be doubled. Okay, chaliye. Everyone, let's talk about the process of replication. Now, what is the process of replication? Of course, in which the DNA will be replicating itself, right? And it needs certain important components. Now, what are the components required by DNA? क्या-क्या चाहिए DNA को? DNA process, पूरा process करने के लिए. Definitely, it requires some enzymes. It requires an origin, right? I'll write origin over here. Basically, in short, the location. Clear? And of course, these are the two important things. Now, in enzymes, there are various enzymes that DNA will be utilizing in the DNA replication ka whole process. We have approximately 6 important enzymes. Everyone, are we clear? We'll be discussing about each of these. But everyone, are you clear with that? They require the enzymes and the origin, the location. Okay, so let's get started. The first step that we have is the Finding of the origin of replication. So again a very important information for all of us. ये जितना भी हम पढ़ रहे हैं DNA replication ये किसमें हो रहा है? सब प्रोकैरियोट्स में पढ़ रहे हैं. Right? We are studying in the prokaryotes. Clear everyone? हमारे इस चैप्टर में राइट भास्कर भी बताएंगे एंजाइम कौन कौन से हैं. हमारे इस पर्टिकुलर चैप्टर में ज्यादातर इंफॉर्मेशन दी है हमें फ्रॉम द पॉइंट ऑफ व्यू ऑफ प्रोकैरियोट्स. ठीक है, चलिए. सो डीएनए रेप्लिकेशन हो रहा है. डीएनए रेप्लिकेशन को स्टार्ट होने के लिए क्या चाहिए? उसे एक ओरिजिन चाहिए. और ओरी व्हाट इज दिस? इट्स नथिंग बट द ओरिजिन. अब रैंडमली डीएनए कहीं से अपने आप को रेप्लिकेट करना स्टार्ट नहीं करेगा. डीएनए को एक ओरिजिन चाहिए, कोई पॉइंट होना चाहिए जहां से वो डीएनए रेप्लिकेशन स्टार्ट कर सकता है. तो ऑफ कोर्स प्रोकैरियोट्स में होता है. इफ दिस इज अ प्रोकैरियोटिक सेल्स राइट वी विल बी एबल टू सी कि हाँ यहाँ पे एक पॉइंट ऑफ ओरिजिन हो सकता है और फिर यहां से इधर भी रेप्लिकेशन हो सकती है, यहाँ पे भी हो सकती है. बट in our case of eukaryotes, of course, DNA बहुत बड़ा है, so we have various places of the origin. But वो origin होना बहुत जरूरी है. Without the origin, right, एक point जहाँ पे एक point है जहाँ से replication start होनी, अगर वो नहीं होगा तो replication नहीं हो पाएगी. Everyone, are we clear that point of origin or the origin is super important? So we know that the process of replication starts at a very special region, which is the ovary. That's right. That's the origin. Okay. Now, here we have, right, this is the origin of replication and this is really very important. Please, you can note down this, right? काफी information है हमारे पास में. Randomly कहीं से भी start नहीं होगा. It needs a very specific point, so that is the reason that we have over here. Now, जैसे ही origin of replication mark हो जाएगा, identify हो जाएगा, we will see that we'll have an entry of very special enzyme. The first enzyme, everyone, that we have to remember over here is the helicase. Which enzyme we are talking about? Helicase. नाम याद रहेगा? The first enzyme that will be coming once the origin of replication point is found out is the helicase. And what is the role of helicase? It will be actually helping in the unzipping the DNA. It will actually help in opening of the DNA. This enzyme will act, right, and it will just open up the space. Clear? यहाँ पे enzyme आ गया है तो यहाँ पे open space हो जाएगी. And this is done with the help of the enzyme helicase. So here we have this. Helicase actually helps to unwind the DNA. And it forms this replication bubble where the replication will take place. It's a very, very important thing. Okay. So what will happen? It will uncoil itself, right? The DNA will uncoil itself, and of course we have the helicase over here. ठीक है, ये unzipping में कौन help कर रहा है? Unwinding में कौन help कर रहा है DNA की? We have the helicase enzyme. The first enzyme is the helicase. ठीक है. अब हम आगे अगर बढ़ेंगे so there are very important thing that we have to remember. Now helicase के अलावा there are very special single binding proteins SSBP. Clear? Everyone, are we clear with this? All of you, quick thumbs up. Very good everyone. Please do keep the enthusiasm really very high. Very good. Clear है सबको? Right? Awesome. So we have SSBP, right? These are the binding protein that will make sure कि unwind फिर से दोबारा coiling ना हो. So ये इस तरीके से आके stick हो जाएंगे. And they will make sure कि unwinding फिर से ना हो. मतलब unwinding तो हो गई है, sorry. Winding फिर से ना हो. है ना? बहुत जरूरी है, वरना DNA का nature क्या है? Wind हो जाता है. Coil हो जाता है. तो हेलिकेस क्या कर रहा है? बोल रहा है कि चलो मैं इसको ओपन कर देता हूं. Right? और फिर यहाँ पे आके SSBP. We have these important things that will come. है ना? Binding protein basically जो हमें assure करेंगे कि DNA की दोबारा से coiling नहीं हो रही है. Once this is done, everyone, we know that we have to move ahead. Before we move ahead, we have to quickly recall कि हमारा DNA का क्या structure था. So we remember that we have nitrogenous bases, we have sugar, we have phosphate, and we have the two anti-parallel DNA strand. One runs from 3' to 5', 5 prime, 3 prime. You can also call that 5' to 3'. This is the opposite. not the opposite per se, anti-parallel strand that we have of the DNA. Now this becomes super important. ये बहुत जरूरी है हमारे लिए समझना कि ये दो एंटी पैरेलल जो स्ट्रैंड्स हैं इनका क्या रोल होगा डीएनए रेप्लिकेशन में. सो लेट्स गेट स्टार्टेड एवरीवन. अच्छा, हमें क्या पता है? हमें पता है कि डीएनए की बैकबोन क्या है? वी हैव दी शुगर एंड फॉस्फेट. कौन सा एंड फ्री है? लेट मी क्विकली रिकॉल. कौन सा एंड फ्री है? कहां पे हम देखेंगे कि एडिशन हो रहा है? जल्दी बताइए. Where the new nucleotides will be added, 3' or 5'? Yes, everyone? जल्दी से 3' राइट. सी हमें पता था कि 3' पे क्या हमारा शुगर है, जो OH ग्रुप है वो फ्री है, राइट. तो जो भी एडिशन होता है वो हम यूजुअली बोलते हैं कि वो कहां हो रहा है हमारा 3' एंड पे. वेरी गुड. वंस वी आर क्लियर विद दिस, अब हमारे लिए बहुत इजी हो जाएगा ये समझना कि कहां से, कौन सी साइड से एडिशन हो रहा है. सो हियर वी हैव एवरीवन द डीएनए स्ट्रैंड, राइट. ये है 5' to 3'. क्लियर. इसका एंटी पैरेलल क्या होगा? कोई मुझे बताएगा? व्हाट विल बी द एंटी पैरेलल ऑफ दिस? जल्दी से. इसका एंटी पैरेलल जो स्टैंड बनेगा उसकी क्या होगी? वेरी गुड. उसे हम क्या बोलेंगे? 5' to 3'. क्लियर. यस, 5' to 3'. इस तरीके से होगा. सो ये अगर हमारा पॉइंट है, राइट, अगर यहां पे है तो हम इस डायरेक्शन में यहां से एडिशन स्टार्ट होगा, राइट. वेरी गुड. अब एक और बहुत इंपॉर्टेंट चीज, very super important cheese, we have the entry of a new enzyme. There are two enzymes that we'll be talking about over here. First, of course, we have RNA primerase, right? Primerase basically. Clear? ये क्या ऐड करेगा? ये ऐड करेगा RNA primer. RNA primer में we have approximately 5 to 10 nucleotide. या फिर बेस पेयर बोल लेंगे. So ये यहां पे आके ऐड कर देगा. ये यहां पे क्यों ऐड कर रहा है? बिकॉज वी हैव वन मोर एंजाइम विच इज द हीरो ऑफ दिस होल प्रोसेस, डीएनए पॉलीमरेज, राइट. हमें याद रहेगा डीएनए पॉलीमरेज. और कौन सा डीएनए पॉलीमरेज है? डीएनए पॉलीमरेज 3. ठीक. सो डीएनए पॉलीमरेज 3 बहुत अच्छा एंजाइम है. इट एक्चुअली प्लेज अ वेरी इंपॉर्टेंट रोल इन द डीएनए रेप्लिकेशन. बट थोड़ा सा उसको पुश चाहिए. थोड़ा सा वो बोलता है कि नहीं मुझे थोड़ा सा हेल्प कर दो, देन आई विल डू इट. सो डीएनए पॉलीमरेज को थोड़ा सा पुश करने के लिए वी हैव एन एंजाइम प्राइमरेज, राइट, जो क्या करता है? एक छोटा सा प्राइमर, आरएनए प्राइमर, एक छोटा सा सेक्शन ऐड कर देता है जहां पे हमारे पास में 6 से 10 न्यूक्लियोटाइड और बेस पेयर्स हैं. क्लियर. जैसे ही ये आरएनए प्राइमर यहाँ पे है, जैसे ही ये यहाँ पे एडिशन है हमारे बेस पेयर्स का, डीएनए पॉलीमरेज बोलता है अच्छा तुमने मेरा थोड़ी सी हेल्प कर दी, अब मैं काम संभाल लूंगा. And this enzyme comes over here and start adding the nucleosides. Clear everyone? Clear है? Clear all of you? Are we clear with it? अब ये यहाँ पे base pairs फिर add करना शुरू कर देता है. Clear? Give me a quick thumbs up everyone. Yes. You can take the screenshot everyone wherever you feel it's necessary. Please do take care. And please do take the screenshot. Yes? Okay, chaliye. Now let's see. So we have this RNA primer, right? So हमें पता है at the right end, जहाँ पे another enzyme we have RNA primerase. ये क्या करेगा? छोटा सा DNA primer, RNA primer add करेगा over here. And what is the RNA primer? It's nothing, but it has the 5 to 10 nucleotides of RNA fragments which are complementary to the DNA. DNA की complementary में it will add few base pairs over there or the nucleotides. So it is a starter, and once that is done, we will see that our DNA polymerase will now come and it will say कि भाई अब मुझ पे काम छोड़ दो, मैं सारा काम संभाल लूंगा. राइट. डीएनए पॉलीमरेज आएगा रेप्लिकेशन बबल में और अपना काम स्टार्ट कर देगा. नाउ इट्स अ वेरी इंपॉर्टेंट थिंग एवरीवन, प्लीज फोकस ओवर हियर. यस, अब हम देखेंगे कि डीएनए पॉलीमरेज एंजाइम क्या है। डीएनए पॉलीमरेज एंजाइम एक ऐसा एंजाइम है जो क्या कर रहा है, जो एंजाइम ध्यान से क्या कर रहा है, पॉलीमराइजेशन कर रहा है, पॉलीमराइजेशन ऑफ दी डीऑक्सोन्यूक्लियोटाइड। क्लियर? वो क्या ऐड कर रहा है, न्यूक्लियोटाइड्स को ऐड कर रहा है, पॉलीमराइजेशन, एडिशन हो रहा है, चेन को बढ़ा रहा है वो। सो डीएनए पॉलीमरेज क्या है, इट्स अ डीएनए डिपेंडेंट डीएनए पॉलीमरेज, डीएनए पे डिपेंडेंट है। अगर डीएनए ने बोला मेरे पास में कोई कोड नहीं है, मेरे पास में कोई बेस पे नहीं, तो डीएनए पॉलीमरेज काम करेगा नहीं करेगा। So the DNA polymerase is dependent on the DNA, and once the RNA primers adds few nucleotides, DNA polymerase tells कि अच्छा अब मेरा काम हो जाएगा, I will do the process and will start adding the nucleotides. Everyone, are we clear with this? सिंपल सी बात है, डीएनए पॉलीमरेज बोल रहा है कि ठीक है अब आपने मेरी थोड़ी हेल्प कर दी, मैं अब पूरा काम कर लूंगा, और वो न्यूक्लियोटाइड्स को अब एडिशन न्यूक्लियोटाइड्स का अब स्टार्ट हो जाएगा। क्लियर? तो कैसे होगा एडिशन, ये भी देखते हैं। ये अगर डीएनए हमारा, राइट, ये अगर डीएनए हमारा, राइट, तो यहां पे अगर यहां पे ऐड हो रहे हैं तो क्या हो रहा है, पहले हमारे पास में क्या होंगे, ट्राइफॉस्फेट। ट्राइफॉस्फेट मींस 3 फॉस्फेट हैं, जो भी हमारे पास में ऐसे अगर हम ये से बना दें, राइट, वी हैव 1 2 3, ठीक है, क्लियर? सो वी हैव दिस नाइट्रोजनस बेसिस, ऑफ कोर्स वी हैव, तो ये 3 फॉस्फेट आ रहे हैं। क्लियर? इसमें से क्या होगा, 2 फॉस्फेट चले जाएंगे, दे विल बी गिविंग अस दी एनर्जी। एनर्जी कहां से आ रही है, इफ द डीएनए पॉलीमरेज इज वर्किंग इट रिक्वायर्स एनर्जी, इज अ वेरी एनर्जी एक्सपेंसिव प्रोसेस। सो ये जो 3 फॉस्फेट थे इसमें से 2 ओके डाटा बाय बाय कर देंगे, क्योंकि हमें दिख रहा है ट्राइफॉस्फेट ऐड हो रहा है, बट हमारे पास में सिर्फ मोनोफॉस्फेट रह जाएगा। एवरीवन, आर वी क्लियर? एनर्जी कहां से मिल रही है, यहां से मिल रही है। सबको क्लियर हुआ ये? जल्दी से बताइए। आई होप दैट ऑल ऑफ यू आर क्लियर कि एनर्जी कहां से मिल रही है, यहां से मिल रही है। वेरी गुड, चलिए, यस, डी, राइट, डीएनटीपीज, डीएनटीपीज, डीऑक्सी न्यूक्लियोटाइड्स, राइट, टी है, चेंज हो जाएगा नाम, सी ओवर हियर, ए है, जी है, टी है और सी है, इस तरीके से नाम, नॉमेनक्लेचर याद रहेगा, प्लीज डू रिमेंबर ऑल ऑफ दिस। अब यहां पे हो गया हमारा डीएनए पॉलीमरेज का काम, हमने समझ लिया कि हाउ द डीएनए पॉलीमरेज इज वर्किंग। What is more important is that DNA dependent DNA polymerase will add new deoxynucleosides triphosphate at 3' पे ऐड करेगा OH ग्रुप पे। ठीक है, हमने अभी पढ़ा था, मैंने आपसे अभी पूछा था। Yes, I just asked you कि कहां पे एडिशन होगा, तो you all said कि मैम 3' पे 3' पे OH पे, है ना, जो फ्री OH था हमारा, उस पे we will see the addition happening. Clear? ये समझ में आ गया सबको? चलिए यहां पे देखें, here we have, see, 3' है, राइट, यहां पे OH जो फ्री OH है वहां पे हम देखेंगे न्यूक्लियोटाइड्स की एंट्रिंग। क्लियर? डन 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 एवरीवन, जल्दी से बताइए। सी यहां पे एडिशंस हो रहे हैं, धीरे धीरे आगे जाएंगे, और इसी तरीके से विल हैव अ कॉम्प्लीमेंट्री बेस पेयर्स, राइट, यहां पे T है, A, T, A, G, C, इस तरीके से we will see that DNA replication will keep on happening. अब यहां पे you can take a screenshot of this, that this is what I was telling you about, कि हमारे पास में क्या है, everyone take a screenshot, we have deoxythymidine phosphate यहां पे है, we have 3 phosphate, we have 1, 2 and 3 phosphate, right? After releasing the energy, we will see the 2 phosphate will be leaving. Okay, tata, bye bye. They are leaving us, and what we are left with? We are left with deoxythymidine monophosphate, right? What we have, mono. Mono means single. एक ही phosphate बचा है हमारे पास में. Okay? Clear everyone? ये समझना थोड़ा सा जरूरी है for us to understand के in what way the DNA polymerase is working. Now this question, right, actually help us to understand that why or what is the working of the DNA polymerase. Clear हो गया ना कि कैसे काम कर रहा है, उसे क्या चाहिए और किस डायरेक्शन में जा रहा है और किस तरीके से वो काम करेगा. सो वंस दिस इज डन, हम ये बोल सकते हैं कि डीएनए पॉलीमरेज काम करता जाएगा. फ्रॉम 5' to 3' वी विल सी इट्स वर्किंग. पॉलीमरेज कीप्स ऑन एडिंग द न्यूक्लियोटाइड्स जब तक वो ओपन, जहां पे वो ओपन है ना, जो रेप्लिकेशन फॉर्क बना था हमारा, क्लियर, जब तक यहां तक नहीं पहुंचता वो कंटिन्यूसली ऐड करता रहेगा न्यूक्लियोटाइड्स, राइट. अब ये जो स्ट्रैंड है हमारा, जो 5' से 3' जा रहा है, जो नया स्ट्रैंड बना है हमारा, उसे हम बोलते हैं लीडिंग स्ट्रैंड, राइट. उसे हम क्या बोलते हैं? लीडिंग स्ट्रैंड. और ये बस एक ही बात है समझना है कि इसमें कोई ब्रेक्स नहीं है, ठीक है, यहां पे कोई ब्रेक्स नहीं है. फ्रॉम 5' to 3' कंटिन्यूस एक स्ट्रैंड बनता है, दैट इज द लीडिंग स्ट्रैंड. इसे हम क्या बोलेंगे? लीडिंग स्ट्रैंड बोलेंगे. द अपर स्ट्रैंड जो कंटिन्यूस है, हम बोलते हैं लीडिंग स्ट्रैंड. अब देयर आर टाइम्स जब जो ऑपोजिट साइड है वो है 3' to 5'. ऑफ कोर्स जो वो 3' to 5' है, उसका ऑपोजिट कॉम्प्लीमेंट्री बनाने के लिए वी इट विल बी मूविंग फ्रॉम 3' to 5'. यस, पर अभी हमने क्या पढ़ा है कि जो पॉलीमरेज है वो वर्क करता है ओनली इन वन पर्टिकुलर डायरेक्शन, फ्रॉम 5' to 3'. एवरीवन क्लियर? समझ में आया आप सबको? यस. तो अब यहाँ पे क्या हो रहा है? पॉलीमरेज बोलता है मैं एक ही डायरेक्शन में जाऊंगा, मैं मुझे ज्यादा काम मत दो, मैं एक ही डायरेक्शन में जाके पूरा काम कर दूंगा, मुझे ऐसे 2-3 जगह पे मत भगाओ. तो मैं 5' to 3' ही जाऊंगा, मुझे इधर से चलाना है इधर से चलाओ, इधर से चलाना है इधर से चलाओ. सो वहां पे क्या होता है? अगेन वी हैव द आरएनए प्राइमर व्हिच इज कमिंग ओवर देयर, एंड वो डीएनए आरएनए के वो अपने थोड़े से न्यूक्लियोटाइड्स ऐड करेंगे. अगेन द सेम प्रोसेस विल हैपन दैट इज हैपनिंग ओवर हियर. उसी सेम तरीके से स्ट्रैंड्स आएंगे, डीएनए पॉलीमरेज आएगा, एडिशन होगा, पर इट्स अ डिसकंटिन्यूअस प्रोसेस. इट्स नॉट अ कंटिन्यूअस प्रोसेस. ये छोटे छोटे फ्रैगमेंट्स बनते जाएंगे, ये एक साथ काम ही नहीं कर सकता. जैसे ही वहां पे पहुंचेगा, अच्छा उसका एक एंड आ गया, नाउ अगेन ही हैज टू, और इट्स नॉट अ ही और इट्स नॉट अ शी, इट्स अ इट. सो अगेन एंजाइम को फिर से पीछे कूद के जाके काम स्टार्ट करना पड़ेगा. क्लियर? ऐसे स्ट्रैंड को जहां पे हमारे पास में बहुत सारे छोटे छोटे फ्रैगमेंट्स हैं, व्हाट वी कॉल दैट फ्रैगमेंट एज? वी कॉल दैट फ्रैगमेंट एज दी ओकाज़की फ्रैगमेंट्स. एब्सोल्युटली करेक्ट. हम इन्हें क्या बोलते हैं? Okazaki fragments, because scientists discovered them, and we know that these fragments are छोटू fragments, right? See over here, यहाँ पे आया, इस डायरेक्शन में चल रहा है, छोटा सा स्ट्रैंड बनाया, done with it. ठीक है, Okazaki fragments बोलते हैं हम इन्हें, और it's a discontinuous process. इसीलिए हम इसे क्या बोलेंगे? Lagging strand. Everyone, are we clear? हम इसे क्या बोलेंगे? We'll call it as a lagging strand. Done? It's a lagging strand that we have. Leading strand, continuous है, lagging strand, break हो रहा है. Yes. So here we have the replication fork, right? ये हमारा क्या है? Replication fork है. अगर आप क्लास 12th में है, if you're writing the board examination, common question asked, and इस चैप्टर से ही कम से कम 10 या 9 क्वेश्चन तो आते ही हैं, right? अगर आपको पता है, अगर आपने PYQs देखे हैं, so from this particular chapter, molecular basis of inheritance से कम 9-10 क्वेश्चन तो बनते हैं. So ये चैप्टर बहुत जरूरी है. Every line of this chapter is super important. So are we clear with this? Everyone, are we clear with this? Okay, चलिए. So here's a quick recap. We have the leading strand which is continuous, right? So हम continuous replication, क्योंकि यहाँ पे कंटिन्यूस रेप्लिकेशन हो रही थी फ्रॉम 5' to 3' because DNA polymerase only work in that particular direction and it is anti-parallel to 3' to 5'. Done. The other strand is the lagging strand or the discontinuous replication is happening over here, or we also call them the Okazaki fragments, right? Because again we have the breaks over here. अब ये जो breaks हैं, right? Can you see these breaks? Right? ये discontinuous हैं. Okazaki fragments here we have. Right? अब ये जो breaks आ रखे हैं, what do you think? It's good. Breaks तो नहीं रहेंगे, है ना? ऐसे कैसे breaks के साथ में ऐसे replication करके भेज देंगे? DNA is very conscious about it, right? So what we'll see? We will see that these breaks, right, that we have all over here will be fixed by a very important enzyme which is the DNA ligase. DNA ligase क्या बोलेगा रे? चिंता की कोई बात नहीं, मैं आ गया हूं. I will be helping you with the joining. DNA ligase आएगा, सभी फ्रैगमेंट्स को ज्वाइन कर देगा. Everyone, are we clear? What is the role of DNA ligase? The important role of the DNA ligase is to fill the gaps that are there in these fragments. Clear all of you? So here हमारे जो breaks हैं, जो gaps हैं, वो भी join हो गए. Enzyme कौन सा है? Ligase. Clear? Ligase enzyme. Everyone, are we clear? जल्दी से all of you. Very good. Okay, very good. Right? So हमने क्या क्या एंजाइम देखे? हमने देखा डीएनए हेलिकेज देखा, right? SSBP देखा है, right? We discussed about them. SSBP, then we talked about RNA polymerase, DNA polymerase, DNA ligase, and इसके बाद में we have some enzymes which are the exonucleases, right? वो कैसे enzymes होते हैं? They will be coming and अगर कुछ भी error है, कोई भी error है, ऐसा तो नहीं है कि DNA जो पॉलीमरेज एकदम बढ़िया से काम कर रहा है, करता है बढ़िया से काम और उसकी स्पीड भी बहुत ज्यादा होती है. कैन यू गेस व्हाट इज द स्पीड ऑफ द डीएनए पॉलीमरेज? जल्दी से बताइए. आई एम श्योर आपने कहीं ना कहीं पे देखा होगा, सुना होगा इसके बारे में. डीएनए पॉलीमरेज 2 जो डीएनए पॉलीमरेज 3 जो है उसकी स्पीड बहुत ज्यादा होती है. यस, जल्दी से बताइए. आई एम श्योर यू ऑल आर अवेयर अबाउट इट. Right? So DNA polymerase जो 3 है, it's a very important enzyme, right? And it actually adds approximately 2000 base pair in a second. So it works really very fastly. बहुत जल्दी काम करता है. तो I'm sure जब इतना जल्दी काम कर रहा है तो कहीं ना कहीं पे errors हो सकते हैं, होते हैं. उनको रेक्टिफाई करने के लिए वी हैव डीएनए पॉलीमरेज 1 एंड 2 जो थोड़ा सा प्रूफ रीडिंग कर देते हैं, अच्छा मिस्टेक्स हुई उसको रेक्टिफाई कर देते हैं. वी हैव एक्सोन्यूक्लियस एंजाइम जो एक्स्ट्रा आरएनए के प्राइमर थे उनको हटा देंगे क्योंकि उनकी कोई जरूरत नहीं है. ठीक है, तो ये सब एंजाइम्स बहुत जरूरी हैं इस इन द होल प्रोसेस ऑफ डीएनए रेप्लिकेशन. एवरीवन, आर वी क्लियर? जल्दी से गिव मी क्विक थम्स अप. बढ़िया, सबके डाउट्स क्लियर हो गए हैं? आपको अच्छे समझ में आ गया है? जो बच्चे अभी आए हैं प्लीज मेक श्योर यू गो एंड वॉच इट अगेन, राइट? एक बार दोबारा से देख लीजिएगा एंड आपको समझ में आ जाएगा. यस, वेरी गुड. चलिए एवरीवन, सो नाउ आई हैव फ्यू क्वेश्चंस फॉर यू. इनको सॉल्व करते हैं, देखते हैं हमें कितना समझ में आया है. हियर वी हैव, चलिए से जल्दी से एवरीवन लेट्स टेक अ लुक एट दीज क्वेश्चन. Here we have DNA replication in lagging strand of most of the eukaryotic organisms is. DNA replication in the lagging strand of most of the eukaryotic organism is what? Conservative, continuous, semi-conservative and discontinuous, conservative and discontinuous or semi-conservative but continuous? Semi-conservative but discontinuous, absolutely correct. हमें पता है कि द लैगिंग स्ट्रैंड में वी नो दैट सेमी कंजर्वेटिव तो है ही नेचर, राइट? डीएनए रेप्लिकेशन में स्पेशली इन यूकैरियोटिक ऑर्गेनिज्म, राइट? वी नो दैट इट्स अ सेमी कंजर्वेटिव नेचर ऑफ द डीएनए एंड इट्स डिसकंटिन्यूअस ओवर हियर. ऑप्शन बी इज द करेक्ट आंसर. वेरी गुड. नेक्स्ट क्वेश्चन एवरीवन. डीएनए पॉलीमरेज 1 इज इन्वॉल्व्ड इन. अच्छा, ये प्रॉपर्ली डिस्कस नहीं हुआ पर मैंने बोला था कि वो हेल्प कर रहा है, राइट? डीएनए पॉलीमरेज प्रूफ रीडिंग में हेल्प करता है. रिमूवल ऑफ, यस, एनी हिंट? अभी नहीं तो अभी पता चल जाएगा. वेरी गुड. सो सी, joining of the Okazaki fragments, definitely it's not a job or the function of the DNA polymerase 1, right? Removal of the RNA primer, definitely it's doing, बिल्कुल वो कर रहा है, right? Removal of the RNA primer and filling of the gaps also. So both A and B. Clear everyone? Both A and B we have the answer. Good. Very good. One more question everyone. A DNA molecule in which both strands have radioactive thymidine, right, is allowed to duplicate in an environment containing non-radioactive thymidine, thymidine, right? what will be the exact number of DNA molecules that contain the radioactive after 3 replications? So हम बात कर रहे हैं थाइमिडीन की, राइट? हमने दो स्ट्रैंड्स थे, एक में हमारे पास में है रेडियो एक्टिव थाइमिडीन, राइट, और दूसरा है उसे दूसरे में डुप्लीकेट किया पर ऐसे एनवायरमेंट में जहां पे रेडियो एक्टिविटी नहीं थी. तो ये पूछ रहे हैं कि आफ्टर 3 रेप्लिकेशन में व्हाट विल बी द रिजल्ट. जल्दी से कितने स्ट्रैंड्स होंगे, व्हाट विल बी द एग्जैक्ट नंबर ऑफ द डीएनए मॉलिक्यूल्स दैट कंटेन द रेडियो एक्टिव थाइमिडीन आफ्टर 3 रेप्लिकेशन. सी अगर आप एक्सपेरिमेंट, वो जो पुराना एक्सपेरिमेंट था जो भी हमने डिस्कस करा, उस पर जाएंगे तो यू विल बी एबल टू फाइंड द आंसर. आई थिंक इट विल टेक सम टाइम, राइट? सो इट्स अ होमवर्क क्वेश्चन एवरीवन, राइट? It's a homework question for all of us. I have one more homework question, but let me just quickly skip this part and over here I have few more questions for you. Right, chaliye everyone, here is a question. In the base sequence of one strand of DNA is GAT, TAG, etc., etc. What shall be the sequence of its complementary strand? It's the easiest question, everyone. हमारे पास ये strand है, एक DNA का ये strand है, इसका complementary strand क्या होगा? G के सामने आएगा C, A के सामने आएगा T, T के सामने आएगा A, right? Yes, T के सामने आएगा we have A, then A के सामने T, G के सामने C, फिर G, फिर T, फिर A, then we have C, T, G. So the option number D, very, very good everyone. So option number D is the correct answer. Very good. Next question everyone. The direction of DNA replication is super easy. ये तो आप ही बता दो. The direction in which the DNA replication happens, in which direction? The direction the DNA replication is, it's based upon the, कैसे होता है? It's based upon the, which enzyme? Very good, DNA polymerase, and of course it's from 5' end to the 3' end. 5 prime, 5 to 3. Good. So option number A is the correct answer. Everyone, this question came in the year 2017. During DNA replication, Okazaki fragments are used to elongate, right? During the DNA replication, Okazaki fragments are used to elongate what? The lagging strand towards the replication fork, the leading strand away from the replication fork, the lagging strand away from the replication fork, or the leading strand towards the replication fork. सोचो थोड़ा सा इसके बारे में. We are talking about the Okazaki fragment. So if I draw the replication fork over here, if this is the leading strand, if this is the lagging strand, right? Okazaki fragments are used to elongate what? Right? यहां से चलते हैं ना वो. So basically we can say that the lagging strand is away from the replication fork. Yes, so option number C is the correct answer. It's a trickier question everyone, but interesting. Next question, which came in the long back, but yeah, let's take a look over here at the question. Semi-conservative replication of DNA was first demonstrated in. Semi-conservative replication of DNA was first. This is very easy. We actually saw the first example, if we have to say, was in which organism? Definitely it's a prokaryotes. Yes. E. coli. Option number C is the correct answer. Good. Very good, everyone. Okay, now let's take a look at the last question and then we will end. I have one interesting homework question for you. The Okazaki fragments in the DNA chain's growth. The Okazaki fragments in the DNA chain growth result in the transcription, polymerization in 3' to 5' direction and from the replication fork. Prove semi-conservative nature of DNA replication or polymerize in the 5' to 3 direction and explain 3' to 5 DNA replication. Yes, option number D. Very good, very good. Polymerizing the 3' to 5' to 3 direction वो इसी तरीके से addition होता है and explain the 3' to 5-DNA replication. So that's option number D, everyone. Okay. So with this, everyone, we are done with our today's class. I hope that all of you have enjoyed today's class. Right, everyone? Yes, I hope that all of you have enjoyed today's class, and I hope that now you have understood what is DNA replication. Yes? Quick thumbs up, everyone. I hope that all of you have enjoyed. Yes? Interesting. So we will be discussing transcription tomorrow. So please make sure you come prepared. एक बार आप NCERT पढ़े ना. Today we don't have the NCERT, but कल हम पढ़ेंगे उसको. DNA replication का कल we will quickly go through it. ठीक है? So now everyone, leaving you with one question that I gave you. आप वहाँ पे one homework question, you can go back and see that and read and watch this video again to understand more about the DNA replication. Right? Thank you so much, everyone. Thank you so much. We'll meet really very soon again. Right? Do take care of yourself and keep on learning with Aakash BYJU'S. Good night, everyone. Good night. Good night to Ma, Medha, Tanu, Captain Lucky. Thank you so much. Thank you so much, everyone. We'll meet tomorrow again with the topic transcription. So, good night, everyone. Bye-bye.